For much of the 20th century, the scientific community operated under a widely accepted, seemingly straightforward understanding of human ancestry. This narrative presented a clean and uncomplicated account. Modern humans first emerged in Africa, approximately 200,000 years ago, embarked on a grand migration out of the continent around 60,000 years ago, and subsequently spread across the globe, gradually populating the rest of the world. According to this established view, every individual alive today, regardless of their geographical location or ethnicity, was understood to be a direct descendant of that singular African exodus. The narrative felt complete. Textbooks had been revised, and the scientific consensus seemed firmly settled. To fully appreciate the revolutionary impact of later findings, it's essential to understand this preceding consensus. The prevailing theory, known as out of Africa, posited that all anatomically modern humans originated in East Africa. From there, they spread across the globe in a solitary migratory wave, completely replacing every other hominid species they encountered along the way. According to this theory, existing hominid populations, such as Neanderthals in Europe and Homo erectus in Asia, were entirely supplanted. The explanation for their disappearance cited intense competition for resources, direct conflict, or unfortunate circumstances. Crucially, this model explicitly denied any interbreeding or genetic exchange between modern humans and these earlier hominid forms. The replacement was considered absolute. This conceptual framework held sway over anthropology for 4 decades. Yet this long-held belief was ultimately incorrect. This foundational understanding was dramatically challenged in 2010, when a pioneering team of geneticists in Leipzig, Germany, achieved an unprecedented feat. The first significant challenge emerged when a team at the Max Planck Institute, spearheaded by Svante Pääbo, successfully extracted ancient DNA from Neanderthal bones, discovered within a cave in Croatia. Recovering viable ancient DNA from bones of such immense age was considered an almost insurmountable challenge. The vast majority of genetic material had long since degraded. Undeterred, Pääbo and his dedicated team innovated new techniques, enabling them to meticulously sequence the bones fragment by fragment. Through this painstaking process, they successfully reconstructed the complete Neanderthal genome from millions of minuscule genetic shards. Upon completion of this monumental task, they possessed something truly unprecedented in scientific history: a complete genetic blueprint of a previously extinct human species. The next crucial step involved a direct comparison. The newly sequenced Neanderthal genome was meticulously aligned against the genomes of 5 contemporary humans, specifically 2 individuals from Africa and 3 from outside the continent. The results were stark. The African individuals exhibited virtually no genetic overlap with the Neanderthal DNA. Conversely, the non-African individuals presented an entirely different picture. Their genomes contained distinct stretches, unmistakable segments of Neanderthal DNA, permanently embedded within their genetic code. The implications of this discovery were staggering. It definitively indicated that sometime between 50,000 and 60,000 years ago, as modern humans migrated out of Africa and ventured into Eurasia, they did not simply supplant the Neanderthal populations. Instead, they interbred. These 2 distinct species, which had evolved separately over hundreds of thousands of years, developing unique physical characteristics, differing brain structures, and distinct immune systems, converged once more. Against previous assumptions, they successfully mated and produced viable offspring. These hybrid offspring thrived, passing on their mixed heritage. Consequently, every European, East Asian, and Native American alive today carries the indelible genetic fingerprint of that ancient encounter. However, the unfolding narrative of human ancestry had only just begun. In 2008, Russian archaeologists made another significant find. In a remote cave located within Siberia's Altai Mountains, a tiny finger bone. This relic, no larger than a child's pinky, had lain buried within the cave floor for an astonishing 40,000 years or more. The The bone was promptly dispatched to the same esteemed laboratory in Leipzig. When the analytical results returned, the scientists were reportedly incredulous. The extracted DNA yielded a startling revelation. It matched neither modern humans nor Neanderthals. It represented an entirely distinct entity, a third hominid species, previously unknown to science, that had coexisted with both modern humans and Neanderthals for hundreds of thousands of years. This newly identified species was christened Denisovans. When researchers compared the Denisovan genome to contemporary human populations, they uncovered an even more astonishing pattern. Individuals hailing from Papua New Guinea, Australia, the Philippines, and various parts of Indonesia were found to carry a remarkable proportion, up to 6%, of Denisovan DNA. Certain isolated populations in Tibet displayed even higher percentages. Various East Asian groups also carry traces of Denisovan ancestry. Initially, this geographical distribution presented a perplexing paradox. The sole fossil evidence of Denisovans had been confined to a single cave in Siberia. How could populations situated 10,000 kilometers away possess such a substantial proportion of their DNA? The realization reshaped our understanding of ancient human migrations. It became evident that Denisovans must have inhabited enormous swaths of the Asian continent, adapting to diverse environments. As modern humans expanded eastward from Africa, they encountered Denisovan populations not merely once, but through multiple interactions across numerous locations over thousands of years. The story becomes even more compelling when investigating specific Denisovan genes. The unique physiological adaptation that allows Tibetans to thrive at extreme high altitudes, environments where oxygen levels are perilously low for most humans, is directly attributed to a Denisovan gene known as EPAS1. Through this ancient interbreeding, modern humans acquired a crucial genetic adaptation that enabled them to successfully inhabit and prosper in the rarefied atmosphere of the Himalayas. Without that pivotal mixing event, contemporary Tibetans would likely be unable to sustain life in their ancestral high-altitude homeland. Concurrently, an equally astonishing narrative was unfolding in Africa. For decades, scientists assumed that African populations represented the purest lineage of modern human ancestry. The rationale was that having remained within Africa, these populations had not encountered Neanderthals or Denisovans. Therefore, their DNA offered a direct, untainted link back to the original Homo sapiens. However, in 2016, this notion was challenged when geneticists analyzing the genomes of West African populations, notably the Yoruba and Mendi, uncovered anomalous segments of DNA that starkly contradicted the prevailing model. These unusual genetic segments did not correspond to any known modern human population. They were too ancient, too genetically divergent. The conclusion was inescapable and profound. Africa itself harbored its own distinct ancient hominid populations, entirely separate from the Neanderthals and Denisovans found in Eurasia. Furthermore, modern humans on the African continent had demonstrably interbred with these indigenous ancient groups. This enigmatic ghost population, as scientists began to refer to it, has never yielded any physical fossil evidence. No bones, skulls, or teeth have ever been identified. Yet, its unmistakable genetic signature is clearly discernible within the DNA of millions of West Africans living today. Remarkably, some estimates indicate that up to 19% of the Yoruba genome originates from this as-yet unidentified ancient species, a significantly higher proportion than the Neanderthal percentage typically found in Europeans. This discovery unequivocally means that the genetic history of Africa is far from simpler than that of Eurasia. In fact, it is demonstrably more intricate. Evidence suggests that at least 2, and potentially 3, distinct hominid species were actively mixing and interbreeding on the African continent for hundreds of thousands of years, even prior to the emergence of anatomically modern humans. The analysis of global DNA yielded results entirely unanticipated by the scientific community. It revealed that every single human population on Earth carries the genetic fingerprints of multiple ancient, often extinct species. The notion of any pure lineage was unequivocally shattered. The boundaries between species, considered absolute, were highly porous. The ramifications for our understanding of race and ancestry are profound. The traditional categories, the distinctions between European, African, and Asian, between white and black, are unequivocally not biological realities. They are deeply ingrained cultural constructions superimposed upon a genetic reality that is infinitely more interconnected. Consider skin color. Historically assumed to represent deep genetic differences, contemporary science paints a different picture. Skin color is regulated by a small cluster of genes that adapt to geographical pressures. The earliest modern humans to inhabit Europe possess dark skin. The evolution of lighter skin tones is a recent phenomenon, occurring within the last 8,000 years, coinciding with agriculture and reduced vitamin D intake. The genetic basis for lighter skin emerged partly from Neanderthal DNA and partly from new mutations. In essence, white skin is a relatively recent evolutionary adaptation, conveying very little about the deeper genetic narrative. This principle extends to virtually every other visible trait. They represent superficial adaptations overlying a genome overwhelmingly shared by all. Approximately 99.9% of the human genome is identical across every population. Of the variation, the overwhelming majority is observed within populations, rather than between them. A randomly selected European often exhibits more genetic variation compared to another European than to an African. This underscores that conventional categories of race bear no meaningful correspondence to biological realities. Real ancestry, the kind inscribed within your DNA, disregards such arbitrary boundaries. Journey back 20,000 years, and your ancestors were nomadic hunter-gatherers, bearing little physical resemblance to you. Asia, too, was not settled by a single wave, but by multiple distinct migratory waves, each creating a unique genetic mosaic that defines modern Asian populations today. And then, perhaps the most astonishing revelation of all emerged. In 2018, while analyzing another minuscule bone fragment from Denisova cave, the very same site where the initial Denisovan finger bone had been unearthed, scientists stumbled upon a discovery that defied all previous predictions. This particular bone belonged to a young girl who lived approximately 90,000 years ago. When her DNA was meticulously sequenced, the results were extraordinary. Her mother was a Neanderthal, and her father was a Denisovan. Her very existence provided irrefutable proof that Neanderthals and Denisovans, despite having been separated by hundreds of thousands of years of independent evolution, were still actively interbreeding and producing viable offspring even deep into the last ice age. And if Neanderthals and Denisovans were interbreeding with each other, and modern humans were simultaneously interbreeding with both of them, then the entire understanding of human evolution transforms into something almost impossible to articulate in simplistic terms. It ceases to resemble a neat, branching tree and instead becomes an intricately tangled web, far more complex than previously imagined. Different populations diverged, only to converge again. They mixed, separated, and subsequently mixed with newly encountered groups, gradually merging into the astonishing genetic patchwork that inherently defines every human alive today. There is no pure lineage to be found. No pristine, original population has survived intact through millennia. Instead, every single human group existing today is the complex product of countless ancient encounters, some of which occurred between species once believed incapable of producing viable offspring. There remains one illuminating chapter. Scientists have begun to identify which specific ancient genes remain active in us. A gene known as STAT2, inherited from Neanderthals, enables modern humans to combat viral infections. As modern humans entered Eurasia, they encountered novel pathogens. Rather than awaiting slow mutations, they acquired advantageous Neanderthal genetic variants directly through interbreeding. A powerful evolutionary shortcut. This genetic exchange proved to be one of the most potent tools for early modern humans. We are all inextricably related. The apparent differences we perceive pale in significance when compared to the vast, ancient inheritance that we universally share in this tangled web of human ancestry.